Good day, everyone. Me and my team will be presenting to you the critic report on the biohydrogen production on dark fermentation of agricultural waste, the novel strain, and feedstock characterization. Now, me and my team consisting of Alvin Putra, Devlin Zaira, Rehan Xavier, Relgan Jabez, Chunkai Wei, and Hytham Pedro Sylvester. Our team has conducted a thorough review on the study's strength, weaknesses, and overall contribution to its field. Now,
Hello, my name is Ethan Pedro. I'm going to give a brief introduction about our topic. Our topic is concerned about the dependence of the world on fossil fuels. This dependence has driven economic growth for many countries, but also led for serious consequences. Has environmental damage, raising emissions, and depletion of finite resources. Today, the need for clean and sustainable energy alternatives is more urgent than ever. So, for this project, we have the solution and the opportunity. For the solution, we found hydrogen, which offers a powerful and clean alternative. And when hydrogen is combusted, it produces only water with zero carbon emissions. This makes hydrogen a key contender in the transition to clean energy. For the opportunity, we found agriculture waste. By agriculture waste for hydrogen production, we can reduce environmental impact, lower dependence on fuels, uh, support rural economies, and produce clean and renewable energy at scale.
So, the next one is research scope and objective. In this paper, they are trying to find out what is the microorganism that fit to create a biohydrogen to produce a biohydrogen efficiently in a dark mechanism or without a light. And next one is the feedstock. So they utilizing a lignocellulosic agricultural waste, which is agricultural waste, and then they use that microorganism to produce a biohydrogen like wheat, hay, willow, and barley. So it can help the microorganism hopefully can help to create a biohydrogen from these feedstocks. And the goal is to identify which one is the best bacterial can produce a biohydrogen from the feedstock that we have, like wheat and hay from the agricultural waste. So, by using a different of microorganism, they will found one of the best microorganism that can efficiently produce a biohydrogen. So the next one is novel strains and feedstocks. So this is the main three bacterial that have tested in the paper. The first one is Salmonella aeruginosus MR1. It's a facultative anaerobe known for method reduction, now test for H2 production. So they have been tested for H2 production without bringing and also need an oxygen because they are an anaerobe microorganism which is anaerobe without oxygen. Next one is Selvibrio japonicus Weda 107. So this is the next microorganism that they use in experiment. So this is capable to degrading a complex polysaccharides found in the plant cell walls. So in the plant, they have a cells, which mean the outer of the layer from the plants. They have cells, and then the cells is containing a polysaccharides. Polysaccharides is a long bond like a polymer, maybe like a polymer, long bond, but not in a form like polymer, which but they have a long chain in the terms of saccharides, and then the celibrio is capable to degrading the polysaccharides in the layer cell of the plant. So this is why they used in the experiment to produce a biohydrogen. The next one is Sorangium cellulosum. What is Sorangium cellulosum? So it's a mycobacterium that have in a plant. It can be it can be degraded by using a Sorangium cellulosum. What is mycobacterium? So mycobacterium is a bacterial that For they have a form like a root, and then they live in a group. So cellulose can be degrading the mycobacterium explore for the direct conversion of the biohydrogen production.
Now we discuss about theoretical framework. In dark fermentation, the light-independent biological conversion of organic substrates into biohydrogen. Unlike photofermentation, it relies on the exorganic breakdown of glucose via glycolysis. The key factor, intracellular redox balancing, is crucial for efficiency. Then in pretreatment, lignocellulosic biomass like straw and willow contains complex lignin bonds resistant to degradation. The solution, chemical pretreatment with strong acid, is required to release soluble carbohydrates for bacterial consumption. The challenge of lignocellulose biomass like straw and willow is rich in cellulose but protected by lignin. Complex bonds make it resistant to microbial degradation.
Moving on to the strengths and weaknesses of the article, on the positive side, the article files a major research gap. It induced a novel stains like the list of names of bacteria for biohydrogen production, supported also by the excellent visual data and the comprehensive background research. But on the negative side, the weakness lies in its practicality. The paper fails to provide any economic data or specific yield values, making it impossible to calculate for a conversion efficiency. Furthermore, while it identifies as a growth inhibitors, it offers no solution to resolve them, leaving the study incomplete for industrial scaling.
Hi, my name is Shankai Wei, and I will be explaining the analysis of results in regards to strain performance when it comes to producing hydrogen. For this study, it has yielded both some expected results as well as unexpected new insights in regards to the strain performance. Not only that, the author successfully explained how each bacterium's metabolic pathway was able to influence its hydrogen yield overall. This research was able to offer valuable preliminary insights in identifying new hydrogen producing bacteria. Through findings, it has been noted that S. onadensis showed the best performance, as it had the highest stability among the strains and having an even higher stability than conventional strains found on the market, with C. japonicus following behind, performing slightly better than the conventional strains while still performing not as high as S. onadensis did, whilst S. cellulosum performed the worst out of the three, underperforming even conventional strains that can be found on the market, as shown as the graph here. It has been noted that S. cellulosum's high instability could be due to organic acid accumulation. However, this was not discussed in detail in the study. The other two strains have been noted to require further improvement, potentially through a mix of optimization of certain culture conditions, mixed culture approaches, as well as genetic modification. The study paper does highlight several needs in order to improve the findings, those being a larger scale experiment, economic feasibility assessments, as well as scale up strategies for industrial application. Not only that, the discussions that can be found in the study have noted down several areas that lack considerable depth. One such issue is that there are no explanations as to why S. cellulosum shows unstable hydrogen yields. Furthermore, there are no discussions on whether organic acid accumulation reduces the overall process efficiency. Lastly, the paper's analysis of external environmental factors which affect biohydrogen production seem to be missing, possibly due to a massive oversight.
My name is Ran Safarpurna, so I'm about to explain the future research directions based on this project. So the first one is the optimization. This part is to investigate mixed culture and genetic engineering to solve inhibitor issues and improve stability. The second part is in the economics. It can conduct pilot scale studies and cost analysis to bridge the gap between lab and industry. And the last but not least part is sustainability. It would perform life cycle assessment or LCA and explore zero waste opportunities like fertilizer and etc. So the conclusion of this study is this study successfully identifies a promising new candidate for biohydrogen production. However, to bridge the gap between laboratory success and industrial decarbonization, future work must focus on process integration, scale up strategies, and economic feasibility.
For our group discussion, our team relies on collaborating with three key phases. First, during the consultation with Dr. Saiful, we made a strategic pivot based on his feedback. We decided to switch to new article to ensure a higher quality analysis. Our second meeting, we distribute the task to confirm everyone's role in it and to fit a new direction, moving on to a new uh, article. And finally, our last and third meeting, we focus on the execution, assigning a video segment and setting the record guideline for the presentation you are now watching.